Today we will be providing a brief introduction to genetics and addressing the progression of our understanding of the gene that determines beak shape in Darwin's finches. One of the first times in history that natural selection was observed and recorded was during Charles Darwin's 1831 to 1836 voyage to the Galapagos Islands, where he observed many varying beak sizes and shapes among the native finches. The beak of each finch species was adapted to its environment and the resources available to the finches, which changed from island to island. This brings about the idea of adaptive radiation, where a specific ancestor reacts to a selective pressure, for example, limited food availability or predation, and ultimately creates several distinct species. Darwin wrote in his diary, "One might really fancy that from an original paucity of birds in this archipelago, one species had been taken and modified for different ends. It's fascinating to think that centuries later, these basic observations and hypotheses were in fact true. Darwin assumed that most closely related finches were those that looked alike. His observations and data were mainly based off the species phenotypes. Much more recently, however, scientists have discovered that relatedness between species is more accurately determined by genetics, where similar characteristics determined by the chromosomes within DNA are passed down from generation to generation in related species. Only recently has Leif Anderson compared the entire DNA sequence and genome data with the 15 species. This led to certain deviations in Darwin's initial discovery. Research helped identify genes responsible for beak morphology and therefore helped reorganize the relationship between species, as can be seen in this modified diagram. As you can see, one deviation from initial observations dealt with the ground finch Geospiza difficilis. Although spread across 6 islands, it actually comprises 3 species. Furthermore, research has also illustrated how genes are responsible for beak morphology. For example, one of these new discoveries was of the gene ALX1, which is involved in facial structure development in all vertebrates. As such, specific genes like ALX1 are indicative of this morphological feature of evolution in vertebrates. Therefore, ALX1 plays an important role in beak development in Darwin's finches. 2 distinct variants were found to match up with beak shape. The medium ground finch had a mixture of the blunt and pointed gene variants. Selective pressure comes into play once more again here. Droughts that resulted in a change in food supply forced the birds to form more pointed beaks that would enable a changed diet, ultimately dealing with the altered circumstances and environment. Recent discoveries by Anderson were a result of a specific methodology. Blood samples were taken from 200 individuals from all 15 species of finches, as well as from 2 outgroup species from Barbados. This was done by capturing finches in mist nets, extracting blood samples, and then releasing the finches. DNA was isolated from the blood, and the bird's entire genome was sequenced. Phylogenetic grouping of species was determined by comparing proportions of allele similarities between different populations of finches. By identifying differences in the ALX1 genes of the blunt and pointed beak haplotypes, the timing of divergence of beak shape between species could be estimated. Results derived from Anderson's efforts were compared to Darwin's predictions and showed many similarities. For example, all but one of the blunt-beaked finches originally observed by Darwin were discovered to all be homozygous for the blunt beak-associated haplotype. Also, almost all sharp-beaked ground finches were homozygous for a pointed beak haplotype. In addition, these genotypic studies have also shown that between the time of Darwin's observations and Anderson's experiments, natural selection and introgressive hybridization have contributed to changes in some of Darwin's original conclusions. It is interesting to see a developing understanding of the finches since the time of Darwin. The diversification of genes determining the beak shape is definitely more comprehensive than Darwin's initial ideas. Although there have been clear deviations, general discoveries are evidently similar over the last 2 centuries. It will be fascinating to witness the evolution of future discoveries, especially concluding what a specific gene controls and whether or not it is the sole parameter that determines a distinctive phenotype.